Okay, so to get this PowerPoint started, we're going to talk about Helicobacter pylori. So this is just going to be a brief overview of what we're going to be looking at today. So in 1983, spirogram negative rods resembling Campylobacters were found in patients with type B gastritis. These organisms were originally classified as Campylobacter, but were subsequently reclassified as a new genus, Helicobacter. So Helicobacters were subsequently subdivided into species that primarily colonize the stomach, which would be considered the gastric Helicobacters, and those that colonize the intestines would be called the interhepatic Helicobacters. So let's get started. So what we're going to cover is listed here. So we're going to cover the physiology and the structure of the organism, a few biochemical tests that are needed if you're in a clinical or a regular microbiology class right now, epidemiology, pathogenesis via virulence factors, clinical diseases and diagnoses and treatments. And on the right here, if you want to pause the video and read this, this is just the discovery of the organism. I'm not going to read all this because it's a little long, but very cool little story here. So to get started, we're going to talk about the physiology and the structure of this bacteria. So there are currently 32 species that have been characterized. This bacteria are gram-negative rods with a spiral morphology. Also, Helicobacter pylori has polar flagellas, which is very useful for motility. And also, H. pylori have a bacillary or spiral shape in young cultures and can assume coxoidal forms in older cultures. All H. pylori are highly motile. They have a corkscrew motility via that flagella I just talked about, and they also produce a very abundance of urease, which is very important. I'll touch on that in just a moment. And then the properties listed above are important for survival and the gastric acids found within the stomach. So the next thing we're going to cover here are going to be biochemical tests. So this organism is microaerophilic. They have growth at 30 to 37 degrees Celsius. They are catalase and oxidase positive, urease positive. They have no growth at 42 degrees, so this is very important if you are in a microbiology class and you are performing these tests. Glucose positive and then hydrogen sulfide negative. So on the right photo here, this would be a positive test for urease, and these two tubes here would be a negative for urease production. So just thought I'd show you that. So the epidemiology of this organism I have listed here. So the infections are very common, particularly in people with a low socioeconomic class, and also they are very prominent or a factor in developing nations. And also humans are the primary reservoirs for this organism. They are also ubiquitous and found worldwide. So it's a worldwide problem, not just in particular places. And it is person-to-person contact, typically through fecal to oral route. So wash those hands, people. And on the right here, I do have a photo of what I was talking about a while ago and 2 slides before. It's a scanning electron micrograph of Helicobacter pylori in a 7-day culture, and the bacillary and coxoidal forms are, with the arrows if you just want to look at the graph, and those are bound to paramagnetic beads used in the magnetic separation. So this would be a test that you would use in a diagnosis. So next is going to be the pathogenesis via virulence factors. So H. pylori is, I would say, limited in its virulence factors, but I do have the major ones listed here. So number one would be the adhesions. So the adhesions would be how the bacteria would adhere to the cell in the stomach, in this case the endothelial cells. So, and also it produces urease, which neutralizes gastric acids with the ammonia produced as the byproduct. Also, they have flagella, and this flagella is a great virulence factor for this organism because it produces of movement through the gastric mucus, and also vacuolating cytotoxin A, and which is a protein that damages the host cell by producing vacuoles. So the clinical diseases of this bacteria. So the diseases are going to be directly related to the site of colonization. And name 3 of the main diseases caused by this organism are going to be gastritis, peptic ulcers, and gastric adenocarcinoma. I hope I said that right. And this is just a photo here showing you what an ulcer would look like within the stomach of an infected person. So moving on, it's going to be the diagnoses. So microscopy is also used in diagnosing this bacteria. A urease breath test. This is very cool. What you do is you just blow into here and it gives you a positive or negative. And the reason why it gives you a positive is because the bacteria, in this case that we're talking about, produces so much urease that you can actually find it when you breathe it out. So I thought that was very interesting, thought I'd share. And also H. pylori antigen test is sensitive and specific and is performed with the stool specimens. So to finish this PowerPoint up, we're going to talk about treatment. So there are multiple regimens that have been evaluated for treatment of H. pylori infections. So you're going to do a combination therapy with a proton pump inhibitor, which would be meprazole and macrolide, which would be clarithromycin, and also a beta-lactam, such as amoxil. You're going to use that for about 2 weeks, and this has been a very successful rate. So thank you for watching, and I hope you learned something. Hope it helps, and see you guys next time. Oh, also here's my references. Thanks.